Thank you, Natalia, for inviting us to this symposium. Today, we'd like to discuss about hypothalamic impairments in glaucoma patients. In glaucoma, there is an ongoing clinical problem where eye pressure decrease is the only clinically modifiable risk factor for glaucoma interventions. However, glaucomatous neurodegeneration is irreversible and it can still occur after lowering eye pressure. So there's actually an unmet need to identify early biomarkers to slow down the disease progression and also more specific targets to decrease the severity of damage caused to the visual system beyond lowering eye pressure alone. So in our laboratory, we have been testing the hypothesis if glaucoma may involve not only the eye, but also the brain's visual system. For example, in the image-forming visual pathway of our glaucoma patients, using the functional magnetic resonance imaging via blood oxygenation level dependent contrast, we can focus on the visual cortex by stimulating upper visual field or the lower visual field using checkerboard stimulation. And we can find that in the visual cortex of the healthy control, across, all the way to early glaucoma and advanced glaucoma, the brain activity would gradually decrease in a topological manner for the upper and lower visual field towards the ventral and dorsal visual cortex, which suggests that perhaps there's a weaker brain functional response with increasing glaucoma severity between the eye and the visual cortex. And not only that, if we plot against the visual field threshold with the blood oxygen level dependent brain activity in the visual cortex, for both upper visual stimulation and lower visual stimulation, we may find there's a tipping point whereby the brain activity may continue to decrease while the visual field remain relatively stable until reaching this point when the visual field threshold would gradually decrease. So this suggests that the brain activity in the visual cortex may also be impaired prior to detectable clinical vision loss in glaucoma, which allows biomarkers for detecting early brain activities in the visual pathway. However, in addition to the image forming visual process, there's actually a non-image forming visual process in the brain. This process connects between the retina and also the hypothalamus and is playing important roles in regulating the autonomic nervous system, such as the intraocular pressure and also the blood pressure. This pathway may also regulate the circadian rhythm as well as energy balance. Of note, in the hypothalamus, there are multiple subunits, and the retina is particularly directly connected to the suprachiasmatic nucleus in the front of the hypothalamus. So we want to ask the research question, does glaucoma affect both the structure and the function of the hypothalamus that may correspond to the non-image forming visual pathway? To test this hypothesis, we recruited 27 healthy controls, 23 early glaucoma patients, as well as 29 advanced glaucoma patients. We used different measuring measures from clinical ophthalmic tests such as visual field perimetry and optical coherence tomography of the retina and optic nerve, followed by anatomical magnetic resonance imaging at 3 Tesla, as well as functional MRI. For functional MRI, during the scans, the flickering checkerboard was presented for 8 seconds on condition, as well as a gray screen called the off condition for 16 seconds in each trial for 22 trials. Because of the resolution of the MRI, we were unable to segment different small subunits of the hypothalamus. So we use the hypothalamus to map and segment into 5 subregions using free server. And then we obtained both the anatomical volumetric size and also the percentage signal change of the blood oxygen level dependent response in the hypothalamic subunits. So in terms of results, we noticed that across the 5 subregions, most of the changes were focused in the ventral or bottom part of the hypothalamus. For example, in the anterior inferior regions, we have the decrease in the volumetric size when the subjects are progressing from healthy towards advanced glaucoma. This also occurs in the tubular inferior regions from healthy to early to advanced group, as well as the posterior region. And not only that, in the anterior inferior regions, the anterior inferior volume is also linked to both the systolic blood pressure as well as the plasmatic blood pressure. For the tubular inferior regions, the volume is also linked to the retinal thickness across the peripapillary retinal nerve fiber layer, as well as the macular ganglion cell in the placiform layer. And the volume is also linked to the visual field mean deviation, as well as the foveal threshold. Last but not least, the visually induced response in our glaucoma patients in the tubular inferior regions also tend to be relatively flattened upon visual stimulation when compared to the healthy controls. In the more dorsal side of the hypothalamus, we don't observe volumetric changes, but the superior tubular regions also have some linkage to the eye pressure. We are also interested to look into a larger data set to validate the current observations. So we attempt preliminarily into the UK Biobank using 400-ish healthy subjects and also 400-ish age and gender matched glaucoma subjects. And we also observe that there is a significantly smaller volume in the hypothalamus of our glaucoma subjects relative to healthy controls. In summary, glaucoma patients exhibit reduced volumes in the tubular inferior and anterior inferior hypothalamic subregions, and these measures correlated with ophthalmic measures and blood pressure. Glaucoma patients also showed stimulation-induced brain activity change in the tubular inferior hypothalamic subregions. These observations suggest altered hypothalamic structure and function in glaucoma patients that may contribute to the dysfunction of autonomic functions, including blood pressure and intraocular pressure. In the future, we are interested in widespread brain changes and their behavioral relevance in glaucoma. For example, there are intrinsically photosensitive retinal ganglion cells projecting to the hypothalamus subregions, and these regions may help regulate sleep quality, which may be impaired in glaucoma. We are also interested in the brain regulations of anxiety and cognitive controls in glaucoma, and our initial study suggests there may be altered functional connectivity between the eye and the hypothalamic that may be playing some roles in these functions. With that, I'd like to thank our lab members in the neuroimaging and visual science laboratory, especially Dr. Ziwan Ban and Justin, who contribute significantly to this study, our volunteers, our collaborators, and also the following funding support. Thank you for your attention.